Hello everyone, I'm Shikha Laloria, Professor of Biochemistry at IISc. Today's lecture is on DNA replication. In the first part of this lecture, we will discuss about the chemical basis for DNA replication, and in the second part, we will discuss the mechanism of DNA replication. This figure shows specific base pairing scheme in the double helix as was described by Watson and Crick. Note that the two strands are running anti-parallel, that is 5' to 3' on the left strand and 5' to 3' this way on the right side. So they are running in opposite directions. The bases in the two anti-parallel strands are complementary to each other following the model of specific base pairing suggested by Watson and Crick, that is A only pairs with T and J only pairs with C. So this model actually suggested a mode for DNA replication or for the copying of the genetic material, as was mentioned in the famous statement by Watson and Crick in their paper proposing the model of DNA structure. They had suggested that the specific base pairing proposed by them immediately suggested a possible copying mechanism for the genetic material. This intrigued many scientists at that time, and many possible models for DNA replication were debated. One possible model was the semi-conservative model for DNA replication, which is illustrated in this slide. The two parallel strands separate. Each serves as a template for the synthesis of a new daughter strand by complementary base pairing. In contrast with the conservative model, which would predict that the entire new DNA duplex would be formed with both newly synthesized strands. Or also in contrast with a dispersive model where new DNA synthesis would start on both the strands at short distances, forming regions of new DNA which are dispersed among the old DNA on both the strands. In order to test the models for DNA replication, two scientists, Messelson and Stahl, did an elegant experiment using the heavy N15 isotope of nitrogen to differentially label the parental strand of DNA to distinguish it from the daughter strands having N14 incorporated after shift to the N14 based medium. This experiment has also been referred to by some as the most beautiful experiment in biology, and indeed it's a very elegant classic experiment that all of you should read about. This experiment made very clear predictions about the results that would support semi-conservative mode of replication versus conservative or dispersive modes of replication. So in semi-conservative mode of replication, the strands separate and each parental strand templates the synthesis of one full new DNA strand. So there is still one parental strand in each daughter molecule. In case of conservative, both the parental strands remain together and form an entirely new DNA molecule that has both new strands. Whereas in case of dispersive, each parental strand is broken at intervals and used to prime synthesis of short new strands that would be joined later on to form completely new strands. All the DNA strands produced would have both old as well as new DNA. So this, which is non-conservative, and fully light would be formed after many generations on N14 in the case of dispersive mode of replication. So here in this figure, bacterial cells, they were grown originally in N15 based medium, where ammonium chloride was the sole nitrogen source. Now, if it was conservative, the heavy light intermediate would never have been seen. After one cycle, we would see heavy and light. So basically, let's review the experiment. So the cells, they were shifted to N14 medium, and at each generation or doubling, the DNA was isolated. And it was mixed with a cesium chloride solution, 6 molar, with a density of 1.7 grams per ml, and this was then centrifuged in an ultracentrifuge at 140,000 G for 48 hours to form a density gradient. Okay, so in this, the higher density would be at the bottom of the tube and the lower density would be near the top of the tube. So looking at the results, the DNA would sediment as per its density. And what was observed was that after one generation, the DNA migrated at a region where there is intermediate density consistent with heavy light DNA, and after 2 generations from the same, one observed heavy light as well as light DNA. And on an additional generation, again heavy light and light was formed. This time there was more of the light DNA being formed. And of course, heavy was never recovered again. So these results were consistent with a semi-conservative mode of DNA replication. Because as we already discussed, if it was dispersive, then although in the first cycle it may look similar to semi-conservative, in the second cycle the light would not appear immediately, but we would observe an intermediate band between the heavy light and the light. And likewise in the third and so on. And fully light in that case would be formed only after many generations. So these results were certainly ruled out, both the dispersive as well as the conservative model. As I already explained, in case of the conservative model, after one generation, you would get heavy and light, and you would not get the intermediate one. And that was, of course, not observed. So these results were consistent with a semi-conservative mode of DNA replication. So let's discuss the chemical basis and the requirements for DNA replication. Deoxyribonucleotide triphosphates are utilized as substrates for addition of new nucleotides to the chain. Also, something known as a primer template junction is required for DNA replication. Certainly you need a primer to prime the synthesis or addition of the next base, and complementary base pairing with the template helps in inserting the correct base in the new DNA strand. DNA synthesis occurs by extending the 3' end of the primer, and the incoming nucleotide gets linked via a phosphodiester bond to the C5' of the deoxyribose part of the incoming nucleotide. The DNA polymerase enzyme is the one that catalyzes the formation of the phosphodiester bond joining the 3' hydroxyl end of the primer to the alpha phosphate at the 5' carbon of the NTP. And in this process, a pyrophosphate is released. All known DNA polymerases use a deoxyribonucleoside 5' triphosphate as a substrate and add the monophosphate building block to the 3' hydroxyl group of a growing DNA chain, or the end of the primer strand. Shown here is the structure of a nucleotide that forms a building block of DNA, DAMP. The basic building block or repeating unit is a nucleotide composed of a base, a sugar and a phosphate. The nucleotide AMP with one phosphate is shown here. It has a phosphate, a 2' deoxyribo sugar and the base adenine. The phosphate is attached at C5'. The base is attached via an NO glycosidic bond at the C1' of the sugar. In a DNA chain, another nucleotide can be added by the formation of a phosphodiester bond at the C3' of the sugar of the first connected to the C5' of the incoming nucleotide. Thus, the polynucleotides have got a free 5' phosphate and a 3' hydroxyl end. For the growth of the chain, the hydroxyl at the 3' position on the terminal nucleotide attacks the linkage between the alpha and the beta phosphate or phosphoryl groups of the incoming precursor DNTP. One nucleotide gets added and the pyrophosphate ion composed of the beta and the gamma phosphates of the substrate is released. This reaction is catalyzed by the enzyme DNA polymerase. In the cell, both the DNA strands are replicated simultaneously. So the two strands of the DNA double helix, they separate to form the two template strands on which the new DNA will be built. And this Y-shaped junction which is there between the two unreplicated DNA and the newly separated template strands is referred to as the replication fork. A multi-enzyme complex that contains the DNA polymerase synthesizes DNA at the replication fork. Now recall that these two DNA strands are anti-parallel, but at the same time DNA synthesis by DNA polymerase occurs only in the 5' to 3' direction. Hence, as a result of this, the replication fork is asymmetrical due to mainly two reasons. One is the anti-parallel strands and also that the polymerase synthesizes only in the 5' to 3' direction. So the DNA polymerase at this replication fork synthesize only in one direction from 5' to 3' going this way, and the DNA polymerase initiates DNA synthesis by adding nucleotides to the 3' end of a primer, and the primer used is actually an RNA primer. The reason being that the DNA polymerase itself, it cannot put together or join two nucleotides by itself. It needs a primer. It needs a 3' hydroxyl in a duplex. Since the two DNA strands are anti-parallel, the DNA on one strand, which is referred to as the lagging strand, which is shown here, has to be synthesized in short segments, and these segments are known as Okazaki fragments, which range from 100 to 2000 bases based on the organism that you're working with. These fragments later, after near the end of the replication, are joined by a DNA ligase. So this was actually found by an interesting experiment done by the scientist Okazaki, after whom these fragments are named. So tritiated thymidine was added to dividing bacteria for a few seconds to label only the most recently synthesized DNA fragments. And DNA fragments that were about 1000 to 2000 nucleotides long were formed transiently. And these were made in the 5' to 3' direction, and they were joined after synthesis and later on incorporated into longer DNA chains. So these short fragments were referred to as Okazaki fragments that we'll be referring to a lot. The direction of polymerase movement on the leading strand is the same as the overall direction of DNA replication. The direction of polymerase movement on the lagging strand is opposite to the overall direction of DNA replication. The Okazaki fragments are made in a sequential order. Those which are nearest to the fork would be the one which has been synthesized most recently, and older ones are further away from the fork. An enzyme, DNA primase, synthesizes the short RNA primers, which could be 10 nucleotides long, using ribo-NTPs as the substrates. This process is shown here, where the primase is putting together the first two nucleotides to start the RNA primer. DNA polymerase, as I mentioned, cannot initiate polymerization de novo. It can only add to the 3' end of an existing nucleotide that is part of a nucleic acid chain base paired to DNA. However, this enzyme, DNA primase, can start a new polynucleotide chain by joining two nucleoside triphosphates together. And this is shown as the first step here. Now, of course, remember that in RNA, instead of thymine, you have uracil. So that's what's been incorporated here. And the primer is being denoted in yellow, and this theme will be followed in many of the future slides and animations. So please bear it in mind that the primer would be labeled yellow. Okay. So after starting this chain, it can synthesize a short chain also in the 5' to 3' direction, and then it stops, and then it makes it available, the 3' end available to the DNA polymerase for further extension, which is shown in the second panel. So the DNA polymerase can add to the new nucleotides to the 3' hydroxyl and then start the DNA synthesis. Now, primase enzyme adds nucleotides to a single-stranded DNA template, and its activity is enhanced when it is bound to another enzyme, the DNA helicase enzyme, at the replication fork. So these properties ensure that it is only active at the replication fork and not anywhere else where there is single-stranded DNA. So the process of replication continues in this way. Now the DNA polymerase can take over and start synthesizing the DNA chain. Because in the end, these RNA primers have to be removed, and there would be a gap created which has to be filled by a DNA polymerase, and then the neighboring segments of DNA have to be ligated at that site. For DNA synthesis to occur, the two DNA strands that are held together by hydrogen bonding between the bases, they need to be separated. So local opening of the DNA helix happens, and this is first followed by synthesis of the RNA primer. DNA synthesis starts at this primer by the DNA polymerase, initiating the leading strand DNA synthesis. Next, additional primers are sequentially laid down for the lagging strand DNA synthesis, as is shown here. And this results in the formation of Okazaki fragments. This structure, in which both the strands have been separated from each other and both of them are now serving as templates for DNA synthesis, is referred to as a replication bubble. So shown here is a generic diagram showing prokaryotic DNA replication. In prokaryotes, the chromosome is circular, and replication initiates at an origin or an initiator sequence. The replication origin binds a sequence-specific DNA binding protein referred to as the initiator protein. The origins also have an easily unwound region, which is AT-rich in nature. So an AT-rich segment is easily unwound relative to other random sequences, because as you know, AT base pairs have only 2 hydrogen bonds as opposed to GC base pairs which have 3 hydrogen bonds. Replication initiates with the help of initiator protein and a primosome. Primosome consists of a DNA helicase and a primase. The DNA helicase unwinds the DNA, and the primase, of course, as explained, lays down a primer. The DNA polymerase synthesizes leading as well as lagging strand DNA, and replication in this, it proceeds bidirectionally. So there are two forks moving away from each other, and they keep on progressing, and new DNA keeps on getting synthesized until at the other end they collide with each other, and then here the primosome is then disassembled, and DNA replication is completed, resulting in the formation of 2 copies of the chromosome, that is 2 daughter chromosomes. So some of the key requirements for DNA replication in prokaryotes are mentioned in this slide. One, as already mentioned, is the initiator, the DNA A protein. It binds to the origin DNA and it melts the AT-rich region. The helicase or the DNA B protein unwinds DNA at the replication fork to generate single-stranded DNA template strands. Primase or the DNA G protein synthesizes the RNA primer for DNA synthesis by DNA polymerase. Also, of course, most importantly, you need the DNA Pol2 holoenzyme that synthesizes DNA. It consists of a clamp loader, which is a complex that loads another protein complex known as a sliding clamp onto a primer template duplex. The tau protein, which connects the clamp loader to the DNA polymerase 3 core, And this also later on contacts and stimulates the helicase. The DNA polymerase III core enzyme, which is of course required for DNA synthesis, and in the holoenzyme there are 3 copies of this, and a sliding clamp which clamps the DNA polymerase III core and onto the DNA, and it enhances its processivity, so it ensures that it doesn't fall off and diffuse away. So this helps in maintaining the efficiency and speed of progression of DNA synthesis. You also need single-strand DNA binding proteins. They are quite important as they bind to single-stranded DNA near the replication fork and prevent the annealing of the single-strand DNA template strands, either with itself or with the other complementary strand. One also needs enzymes, DNA Pol1, and in some systems there's a different enzyme that does this job of primer removal, and a ligase which is required for ligation of the Okazaki fragments in the end, in the lagging strand. One also needs a class of enzymes known as topoisomerases. In E. coli they are known as topo1 and gyrase, and the main function of these enzymes is that they prevent DNA tangling during replication, and they remove supercoils that are formed because of the progression of the replication forks. Although the preceding descriptions I gave are quite simple, DNA replication is a highly complex process which is brought about by coordinated action of the replisome. The replisome is a complex molecular machine that consists of various enzymes and other proteins which are important for replication, and these are assembled at the replication fork. You can see some of the proteins here doing the process of replication. Replication in E. coli happens quite fast. The DNA polymerase at the fork can add nucleotides at the rate of up to 1000 per second. It's a very complex and dynamic process, and this replisome machine has got many moving parts, as you can see. There are additional regulatory modules also involved in checkpoint control, etc., which are not shown here. The helicase enzyme is shown in blue over here, which is unwinding the DNA duplex. You can see the continuous synthesis on the leading strand below. I'm playing it again to explain this complicated animation, which was provided to me by Drew Berry and Etsuko Ono. And so here you can see the helicase, which was this blue enzyme over there. Yeah, it's unwinding the DNA into 2 single strands, and this side on the right side is the leading strand, and on the top is the lagging strand, which is forming these loops and moving around a lot. So you can see actually the primase, just now it arrived and it formed this yellow primer, and very quickly the action happened where the clamp loader attached the clamp to it, and the DNA polymerase, the lagging strand DNA polymerase, became attached to it. So just watch this a little more to make it clearer. I think we have covered that entire animation. So here you can see this loop which increases in size, and that's seen on the lagging strand only, and this model of replication is referred to as a trombone model, after a musical instrument where you see such an increasing size of one of the parts of the instrument. So here's a helicase, there's the yellow primer and the clamp loader. So this is indeed fascinating process, and in fact a lot of problems can also happen when there's such a complex process going on that may need to be corrected or monitored by the checkpoints. There again, that's the lagging strand, let go of the polymerase. So I think it would be clear. So this very complex process, of course, the key enzyme here is the DNA polymerase enzyme. And I just want to review some of the main properties of the DNA polymerase, which is important for this synthesis reaction in cells. The DNA polymerases, they synthesize DNA by adding a nucleotide to the 3' hydroxyl of a primer. The DNA polymerase enzyme, it's got a single active site that is capable of catalyzing DNA synthesis. DNA polymerase only incorporates correctly paired bases as specified by the complementary sequence of the template strand. Incorrectly paired bases are not incorporated by this enzyme. And the structure of a DNA polymerase enzyme resembles a partially closed hand, and the DNA fits in a large cleft in that molecule. Also, DNA polymerases are processive enzymes, and the E. coli DNA polymerase, DNA Pol 3, which is the replicative polymerase, can add up to 1,000 nucleotides per second. And of course, as I already explained, this processivity is enhanced by being attached to the sliding clamp, which keeps it in place attached to the DNA strand. So even if it were to get disturbed or fall off, the sliding clamp puts it right back to do its job, and it prevents it from floating away, which it would do if it was not tethered to the clamp. DNA polymerases also have a 3' to 5' proofreading exonuclease activity that removes unpaired or mispaired bases. So listed here are a couple of references pertaining to the Messerschmitt-Stahl experiment that had proved the semi-conservative nature of DNA replication. Worth looking into. Stay tuned for the next part of this lecture, which will be on the mechanism of DNA replication. Thank you.